The overall goal of this procedure is to sequence 16S rRNA genes amplified from environmental samples using a benchtop sequencing machine. This is accomplished by first amplifying the 16S rRNA gene using modified primers. The second step is to purify the amplification products, quantify them, and pool them. Next, the Amplicon pool is attached to sequencing spheres amplified clonally during emulsion PCR and sequenced on a benchtop sequencer. The final step is to analyze the resulting sequence datasets. Ultimately, 16S rRNA gene amplicon sequencing is used to determine the composition and diversity of the microbial community in an environmental sample.
The main advantage of this technique over existing methods like DGG or cloning sequencing is that it gives a deeper and clearer perspective into microbial community composition and diversity.
This method can help you answer one of the key questions in microbial ecology. Who's there?
To begin, thaw the forward and reverse primers, the 2x hot start tack plus master mix, and the samples. The sequence of the forward and reverse primers fit the template shown. The sequence contains ion torrent specific adapters, a sequencing key for calibrating the signal intensity at the beginning of the sequencing run, a template-specific primer, and a 10 base pair barcode. Then mix the primer.